Biology, topic 3.1, genes.4. Alleles differ from each other by only one or a few bases. Here we'll be talking about how alleles are different by a few bases. And in this case, the bases are A, T, C, and G. And these are the bases we talk about when we talk about DNA. We'll first be looking at how different alleles of one gene have slight base variations. So here, a gene, meaning a length of DNA, and this length of DNA is hundreds and thousands of these A, T, C, G bases, may have slight differences. So if a gene would normally go A, T, T, C, G, if we had a slight variation where instead of an A we had a T, or instead of a C we had a G, this would be an allele. Although if instead of, say, this A we had the T and this T was still an A, this would still be a different allele. But if it worked, it would still be the same gene. And for quick reference, A, adenine, T, thymine, C, cytosine, and G, guanine. Now, you may be thinking, how can there be differences in a single gene without this being a mutation that completely, messes everything up and makes everything not work? So for this, we have single nucleotide polymorphisms, SNPs, or SNPs. And a SNP is a position on a gene where there can be more than one base. And this doesn't mean that there's a single position where there, for example, are two A's stacked on top of each other. What this means is if there's, let's say, an AAATT, for example, in this string of bases on a gene, and there's a single position, so let's say this position, where instead of the A, we can have a T, and it still functions properly, well, this is a SNP. And the next part of this is that one gene may have, this doesn't mean it will have, it may have more than one SNP. And this simply means that any gene may have one, many, or none at all positions on it where there can be these differences. And this difference, as said before, simply means that there will be an allele instead of the single gene. And so if you have a T instead of an A in this position, on a different allele you'll have maybe a C or a G in a different position. Well, if they still work, and I'll put work in quotes, if it still works, it's still an allele, it means that this is simply an area where there's a SNP and everything still functions. But in the end, we have to remember that even though there are these SNPs, every single allele still has to be similar enough to the original gene that it will still function simply as a variation of that gene. And I'll write this down as a separate point. Every allele of a gene only differs by one or a few bases. So this isn't a situation where we're going to have a gene and every second, or every third position is going to be different and it's still going to be the same gene or an allele of that gene. What we're talking about is there will be a few SNPs, or maybe just one, across the gene where there will be these differences and it will still work as an allele.